Hello everyone, welcome to today's live broadcast, Dopamine System Dysregulation in the Pathophysiology of Schizophrenia and Depression. I am Judy O'Rourke and I'll be your moderator for today's event. We have a few important announcements before we begin. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you are interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Anthony A. Grace. Dr. Anthony Grace is a distinguished professor of neuroscience and a professor of psychiatry and psychology at the University of Pittsburgh in Pittsburgh, Pennsylvania. He received his PhD from Yale University School of Medicine, Dr. Benjamin S. Bunny, and had postdoctoral training with Dr. Rodolfo Linus in the Department of Physiology and Biophysics at New York University School of Medicine. Dr. Grace has been involved in translational research related to the dopamine system for over 30 years. His early work pioneered the mode of action of antipsychotic drugs and the identification and characterization of dopamine-containing neurons, and was the first to provide a means to quantify their activity state and pattern in a way that is the standard in the literature. His current work involves novel treatments for schizophrenia and its prevention, the role of dopamine in anhedonia and affective disorders, and the mode of action of ketamine and novel antidepressant drugs. Dr. Grace has received numerous awards for his research. I will now turn it over to Dr. Grace for his presentation.
Thank you very much, Judy, and welcome all to this virtual conference. What I'll be talking about is some of the basic work that we've been doing looking at the dopamine system and its involvement in different psychiatric conditions, especially looking at the regulation of the system. Now, this is a picture of the dopaminergic system. It consists of the ventral tegmental area, that's this medial area, and the substantia nigra. We know that this area is involved in a number of disorders, including Parkinson's disease, schizophrenia, and drug abuse, and there's emerging evidence that it also has a large involvement in depression. Now, in order to try to understand the system, what we want to do is to be able to record from neurons in the midbrain of an anesthetized rat to understand how these systems are regulated and how they may be dysregulated in disease. Now, how can we record the activity across this population of neurons? Well, we know from previous studies that there are a number of neurons that are firing spontaneously, but also a number of neurons that are quiescent and not firing. The ones that are firing at different firing rates and firing patterns. So what we would like to do is to find out exactly what the population activity in these neurons are. To do this, what we do is we pass an electrode through the substantia nigra in a preset pattern and count the number of neurons that are active per electrode track. This gives us an idea of the population activity, or just how many neurons are active. We then look at the neurons that are active and see how fast they're firing, and also what their firing pattern is. When we look at firing pattern, what we find are individual neurons fire at different types of patterns. In the baseline state, they fire in the slow, irregular pop-pop-pop type of firing pattern. But when the animal is exposed to a behaviorally salient stimulus, dopaminergic neurons fire bursts of action potentials. These bursts of action potentials are thought to be the behaviorally relevant output of the dopaminergic system, and they basically alert the brain that something important has happened. This burst firing is controlled by a brainstem area that's known as the pedunculopontine tegmentum. This provides a glutamatergic input to the dopaminergic neurons in the ventral tegmental area. If we activate this by injecting a GABA antagonist bicuculin to activate this area, what we find is a selective increase in burst firing without getting a change in the number of neurons firing or their firing rate. Now, this is consistent with what we know about the system. The glutamatergic input that comes in activates NMDA receptors. But for NMDA receptors to activate a neuron, the neuron has to be spontaneously firing. If it's not spontaneously firing, there's a magnesium block of the NMDA channel and it doesn't change state, which means only the firing neurons will burst fire. Now, what regulates how many neurons are firing? Well, this is regulated by a different part of the brain, an area called the ventral pallidum. The ventral pallidum is the ventral part of the globus pallidus. It provides a very potent GABAergic input to these dopaminergic neurons. And it also fires in high frequencies. What we believe is that this area holds subsets of these neurons in the silent inactive state. But if we inactivate this inhibitory input, this removes the inhibition and allows the neurons to start firing. Now, what's the relevance as far as the number of neurons firing? Well, this tonic activity controls the level of amplification. So in other words, the ventral pallidum controls the number firing, the pedunculopontine controls burst firing being the signal or the behaviorally relevant output. But the number firing is the gain. So the more neurons that are firing, the more neurons the pedunculopontine can make burst fire. This system, we think, plays a major role in the psychotic symptoms of schizophrenia. Schizophrenia is a thought disorder comprised of three major symptom classes: positive symptoms, negative symptoms, and cognitive symptoms. The positive or the psychotic symptoms have the greatest association with an overactive dopamine system. We've used an animal model based on a disruption of neuronal development with a compound called MAM, or methylase oxymethyl acetate. This is a DNA methylating agent that we find when we give to pregnant rats at gestational day 17 will cause a disruption in the system that when the rats reach adulthood mimic a lot of the things that we can measure in an animal that correlates with what we see in schizophrenia patients. Now, we've published already the number of characteristics of these animal models that are consistent with what we see in humans. So as far as anatomical evidence, we see a thinning of the limbic cortical structures and increased cell packing density. Basically the same structures and the same changes that we see in human schizophrenia brains post-mortem. We also see deficits in behavior. We see deficits related to cognitive symptoms, such as extra-dimensional shift, deficits in negative symptoms, such as social interaction and latent inhibition, and also deficits related to sensory systems, such as impairment in pre-pulse inhibition of startle. We also find pharmacological evidence, including increased responses to PCP, which has also been reported in schizophrenia patients, and increased locomotor responses to amphetamine. Now, interestingly, we see this in the adult, but we don't see it when the animal's in the prepubertal stage, which is consistent with schizophrenia positive symptoms occurring in late adolescence, early adulthood, but not in prepuberty. Now, this increased amphetamine response is consistent with the role for dopamine in schizophrenia. There's a plethora of evidence to suggest that dopamine has an involvement in schizophrenia. All antipsychotic drugs that are in use today work on dopamine receptors and are primarily dopamine antagonists. Also, antipsychotic drugs, amphetamine and L-DOPA, things that increase dopamine release, tend to exacerbate or worsen psychosis in schizophrenia patients and can lead to schizophrenia symptoms in normal individuals. And imaging studies revealed an increased amphetamine-induced dopamine release in schizophrenia that correlates with psychosis. However, despite years of search, There's not evidence for a primary deficit in the dopamine system itself. Instead, we now believe the dopamine system is probably normal, but is dysregulated by other structures, primarily among these the hippocampus. So in the MAM model of schizophrenia, there's a hyperresponsivity of the dopamine system analogous to that observed in schizophrenia patients. And current clinical data shows that there's hyperactivity in the hippocampus, especially the ventral hippocampus of rats, which correlates with the anterior hippocampus of humans. It corresponds with the psychotic state of schizophrenia. Now, is the hippocampus hyperactive in the MAM animal, as imaging studies have shown in schizophrenia patients? Well, when we look at firing rate, we find that the ventral hippocampus is firing significantly faster in the MAM animals. But firing rate's a rather simplistic measure of activity. We know that the hippocampus also responds based on different frequency rhythms. And what we find when we analyze the rhythms is a decrease selectively in the very low frequency 0 to 4 hertz rhythms that are important for functional gating, and an increase in the higher frequency rhythms, suggesting that we're losing this gated output and instead getting a high frequency noise coming out of the system. Now, does this also affect dopaminergic neuron activity? Well, when we look at our 3 parameters of dopaminergic activity, number of neurons firing, firing rate, and firing pattern, what we find is a selective increase in the number of neurons firing, more than doubling in the number of neurons, without changes in firing rate or firing pattern. Now, the increased number of dopaminergic neurons firing is consistent with what's seen clinically. Studies by Oliver Howes and Philip McGuire in the UK showed that in schizophrenia patients, there's an increase in fluorodopa-labeled terminals of the dopaminergic system in the striatum. Now, fluorodopa labels active terminals, and the increased number of terminals that they see is consistent with the increased number of dopaminergic neurons firing that we see in the MAM rat. So when the hippocampus is hyperactive, it drives the ventral striatum nucleus accumbens, which then inhibits the ventral pallidum and causes this number of dopaminergic neurons firing to increase. We know that this is driven by the hippocampus because if we inactivate the hippocampus in normal animals, we don't see much of a change. But in the MAM animals, it brings dopaminergic firing right back down to normal without changing the other parameters, suggesting that it's the hippocampal hyperactivity that's driving this increased number of dopaminergic neurons firing, which again is consistent with hippocampal hyperactivity in the human schizophrenia patient, leading to this increased number of dopaminergic terminals active. Now, does this change in dopaminergic firing translate to behavior? Well, in the normal animal, if we give a dose of amphetamine, we get an increased locomotor behavior. In the MAM animal, what we find is the increase in locomotion is substantially greater than what we see in controls. But if we inactivate the ventral hippocampus, something that brings dopaminergic firing back to normal, we also bring the behavioral response to amphetamine back to normal. We don't eliminate it, we just bring it back to what we see in controls. So when hippocampal overdrive seems to lead to increased dopaminergic neuron population activity, which we think is the basis for this hyperresponsivity of the dopamine system in schizophrenia. Now, how does the increase in dopamine population activity affect the response of the dopamine system to behaviorally salient stimuli? Well, what we think is that if an animal or if a person is in a benign context, you're sitting at home in a safe neighborhood behind locked doors, the hippocampus has the dopamine system adjusted very low. Not a lot of dopaminergic neurons firing. Now, whenever a signal comes in, you hear a noise outside, it'll activate the pedunculopontine tegmentum, which will then activate dopaminergic neurons into burst firing. But because there aren't a lot firing, the dopamine response is rather low. So you'll turn around, see what the noise was, and then calmly go back to what you were doing before. But if you're in an activating context, you're an animal out hunting, where every noise is something that you could eat or something that might cause you harm, In this case, the hippocampus has the dopaminergic system toned up very high, a lot of neurons firing. Now the same noise is going to cause a much greater number of dopaminergic neurons to burst fire and a much greater dopamine response. You're going to be on edge, ready to rapidly respond and orient towards the stimulus to identify what it is. Now, what about the case of the schizophrenia patient? In this case, the hippocampus is hyperactive. This causes the dopaminergic system to have a large number of neurons firing spontaneously, essentially every neuron. Because of this, any stimulus that comes in, salient or not, is going to cause a massive dopamine release, which means the schizophrenia patients aren't going to be able to select between behaviorally salient stimuli and irrelevant stimuli, because all of them are going to be causing maximal dopamine activation and require maximum attention as if they're a very threatening stimulus to the patient, which is exactly what they report, the inability to screen out what's relevant from what's irrelevant. Importantly, it also means that the system's going to be activated by stimuli that normally could be ignored. So for example, if one looks out the window and sees two complete strangers talking to each other, this is something that most of us would completely ignore. But to the schizophrenia patient, the dopamine system is going to be screaming at their brain that this is something really important for our survival, which means they're going to have to figure out why these two strangers have such a strong emotional response. And they're going to confabulate that this is the CIA spying on them, these are aliens planning to abduct them. This kind of aberrant salience that Satish Kapoor has talked about with respect to the improper attribution of importance to irrelevant stimuli characteristic of schizophrenia. So when the hippocampus is hyperactive, the dopamine system is hyperresponsive to stimuli and results in overinterpretation of events. So these data show in the MAM model of schizophrenia, the ventral hippocampal hyperactivity leads to a hyperresponsive dopamine system that we think underlies the psychotic state. Why is the ventral hippocampus hyperactive? Well, the most consistent finding that people have made in schizophrenia patients is a substantial decrease of nearly 30% in a specific type of GABAergic inhibitory interneuron, those that contain the peptide parvalbumin. This is found to be decreased across the hippocampus selectively in schizophrenia patients compared to other types of individuals. Similarly, in the MAM animal, we find decreases in parvalbumin neurons in the hippocampus and in the prefrontal cortex, just as been reported by David Lewis and others in schizophrenia patients. So the MAM rats show a regionally selective reduction in these GABAergic inhibitory parvalbumin interneurons. So evidence suggests that both in schizophrenia and the MAM model, there's hyperactivity in the hippocampus due to this decreased interneuron function. And if we inactivate the ventral hippocampus, we can restore normal dopamine system function. Now, how can we fix this pathological increase in dopamine activity to treat schizophrenia? Well, current antipsychotic drugs act by blocking dopamine receptors in the striatum and by inactivating dopaminergic neuron firing. However, the site of action of antipsychotic drugs doesn't correspond to the pathophysiology of schizophrenia. These drugs were discovered by serendipity rather than by a focused effort to try to treat schizophrenia. So although we believe the deficit is in this interneuron pyramidal neuron interaction in the hippocampus, where we treat schizophrenia is actually by blocking dopaminergic receptors in the striatum, which is 1, 2, 3, 4, 5 synapses downstream from where we think the problem is, which should come as no surprise that these drugs probably aren't as effective as we would like them to be. A better approach would be to treat at the site of pathology. We think the site of pathology, again, is at this parvalbumin pyramidal neuron interaction in the hippocampus. Parvalbumin interneurons are driven by NMDA receptors, and in fact, this is where we think drugs like PCP act to worsen the symptoms of schizophrenia because they decrease the activity of the already diminished parvalbumin interneurons. What we would like to do is potentiate the effects of GABA at this synapse. But GABA is difficult to manipulate because it's more than a third of the synapses in the brain. And one can't non-selectively potentiate it like with a benzodiazepine like diazepam because this is very highly sedating. However, nature has given us a handle on this, in that in the hippocampus, there's a selective increase in a GABA-A receptor subtype that contains a certain subunit. Now, benzodiazepines bind to GABA receptors at the alpha-5 subunit, or at the alpha subunit. And there are 6 distinct alpha subunits, but only the alpha-5 subunit is concentrated in the hippocampus. So what we wanted to do is to get a selective GABA-A benzodiazepine or positive allosteric modulator, which we obtained from Jim Cook in Madison, Wisconsin. And what we find is that whenever we use the selective GABAergic compound, we do get selective effects. We can get this decrease in the activation of the entorhinal cortex to hippocampal pathway in the hippocampus by stimulating the entorhinal cortex. But in another area, looking at the cingulate cortex, the thalamic evoked response in the cingulate cortex is not effective even at 10 times this dose. So what we see is a rather selective effect in the hippocampus. When we look at this GABA-A alpha-5 positive allosteric modulator on dopaminergic neuron firing, we also see a selective effect. It has no effect in control animals, but in the MAM-treated animals, it brings dopaminergic neuron firing right back down to baseline levels. Furthermore, whenever we look at behavioral effects, although the effects of amphetamine-induced locomotion are rather variable in the saline animal, in the MAM animal, it brings the hyperlocomotion right back down to baseline levels. So what we're doing is we're actually restoring both dopaminergic neuron firing as well as the behavioral response to amphetamine by this hippocampal selected drug. Now, why isn't this drug used in the clinic? Well, the concern is that there are a number of drugs that have showed promise in preclinical studies that have failed in the clinic. A potential issue is the difference in the schizophrenia patient. Although it's been suggested that the reason these drugs have failed is because we can't translate from animal studies to humans, a concern is that there's a big difference in the way schizophrenia patients are treated. They've basically been given antipsychotic drugs for decades and then withdrawn for 1 week before the drug is tested. And we know that dopaminergic blockers change the system. They basically make the system supersensitive. So what we did is to test the impact of antipsychotic drug treatment and withdrawal on the MAM model. And what we find is that unlike the normal case, where we don't pre-treat the animal and get therapeutic actions, if we look at a rat that has been treated with haloperidol for 3 weeks, withdrawn for 1 week, now we find that our GABA-A alpha-5 compound has no effect. In fact, inactivation of the hippocampus has no effect. And with respect to behavior, what we find is that the animals are still hyper-responsive to the effects of amphetamine, despite the alpha-5 compound or inactivation of the ventral hippocampus. Essentially, the antipsychotic drugs have changed the system from a hippocampal overdriven dopamine system to a postsynaptic supersensitive dopamine system. And this is, again, showing this increased locomotion with haloperidol pretreatment, but not with the pretreatment. So haloperidol or antipsychotic drug pretreatment alters the capacity of the system to respond to a novel agent. And as long as clinical trials are done in chronic schizophrenia patients, it's not clear that we're going to be able to get these novel compounds that may have tremendous efficacy to show actions in these kinds of clinical trials. So in schizophrenia in the MAM model, interneuron dysfunction may be a primary drive of symptomatology. And if we can restore interneuron function within these circuits, we could have an effective therapeutic strategy. The thing is, schizophrenia we know is bad for the brain and is quite stressful. A better approach would be if we could prevent the transition to schizophrenia to begin with. We know that stress plays a major role across psychiatric disorders. Stressful stimuli in schizophrenia patients exacerbate the positive symptoms of this disorder. And we know from studies by Eve Johnstone, Robin Murray, and others that stress can play a role in schizophrenia. Eve Johnstone did a prospective study looking at a number of children that were at risk for schizophrenia and did a number of psychometric tests on them and then followed them to see who transitioned to schizophrenia. And what she found are the children that were hyper-responsive to stress tended to be the ones that converted to schizophrenia. We also know that stress can lead to parvalbumin interneuron loss in the hippocampus, which are the same neurons that are lost in schizophrenia patients and in the MAM rats. So we believe in normal individuals, areas like the prefrontal cortex can limit the response to stress. But if the prefrontal cortex is in a deficit state, we think this can initiate a cascade of events that magnify the effects of stress and lead to interneuron loss in schizophrenia in adults. So in the normal animal, stress activates mesocortical dopamine release, which activates the prefrontal cortex and feeds back and inhibits stress responses. But if there's a deficit, either in the dopamine input to the prefrontal cortex, as David Lewis's data suggests, or in the different regulations of the dopamine system, this is going to lead to increased stress responses. The inability to activate the prefrontal cortex is going to remove prefrontal cortical inhibition of the amygdala, which is going to magnify stress responses. This is going to lead to activation of the noradrenergic system in the locus coeruleus, which is going to magnify stress responses. This will lead to glucocorticoid release, hippocampal damage, leading to magnification of the stress response in this reentrant loop, where a stressor is going to have magnified effects and magnified pathology. Now, are MAM rats more sensitive to stressors, as is proposed to occur in the premorbid state in schizophrenia? Well, what we find are that the MAM-treated rats are more responsive to stress. Mild foot shocks induce focalization in control animals, but the level of vocalization is much larger and longer duration in the MAM-treated animals. Furthermore, if we look at a measure of anxiety, the number of times rats explore an open arm compared to a closed arm, we find that the MAM rats tend to stay in the protected environment, which is consistent with anxiety-like behavior. So if stress plays a role in the developmental etiology of schizophrenia, this suggests that we can intervene early on, we can help prevent the transition to psychosis. We tested this in our MAM model by administering the anti-anxiety agent diazepam peripubertally. So we basically gave the rats either MAM or saline during gestation, then for 10 days around puberty, gave them diazepam. We withdrew the diazepam, waited for them to reach adulthood, and then tested them. And what we found is that the MAM rats that were treated peripubertally with diazepam, the anti-anxiety agent, showed a normalization of dopaminergic neuron firing in the adult. Similarly, they show a normalization in amphetamine-induced hyperlocomotion, suggesting that controlling these stressors early on in life, even in an animal that's been genetically predisposed because of MAM treatment, can prevent the transition to psychosis. So converging evidence from clinical and basic science studies suggests the inability to regulate stress early in life can lead to pathological changes in the hippocampus that may underlie the emergence of schizophrenia in the adult. But if we can identify susceptible individuals and intervene early enough, basically identifying people at genetic risk and seeing if they're hyperresponsive to stress, just by treating the stress, not necessarily with drugs, but with psychosocial intervention, we might be able to prevent the transition to schizophrenia. Now, another disorder in which the dopamine system is believed to play a role is major depressive disorder. Major depressive disorder is a devastating illness that affects as much as 15% of the population throughout their lives. It's a chronic, pervasive disorder responsible for enormous loss of quality of life and societal function in affected individuals. The thing is the antidepressant drugs we have today aren't as efficacious as they would like, and at least 30% of the patients aren't responsive at all. Furthermore, it takes 4 to 6 weeks to get therapeutic actions. These drugs work on the serotonin system, but just like antipsychotic drugs, the use and discovery of these drugs was more by serendipity than it was because of any kind of intelligent design. But we know that the dopamine system actually plays a role in a number of things we see in depression. Multiple studies have shown that if you attenuate the dopamine system, you can get a state of anhedonia, or an inability to derive pleasure from the environment, which is a primary characteristic of depression, as is behavioral despair, that's also seen with dopaminergic inhibition and amotivation. The dopamine system, and we also examined the role of the dopamine system in a model of depression. The model of depression that we used is something called the chronic mild stress model. This is where we give repeated mild stressors, nothing very extreme, things like having a wetting of the bedding one day, having the cage tilted another day, having the lights on for 24 hours the next day, basically chronic unpredictable irritations that have been shown in a number of studies to lead to depressive-like behaviors in animal models. What we find when we expose rats to these chronic mild stressors is an increase in behavioral despair in the forced swim test. This is where rats are placed in a tub of water, and we measure how long they spend searching for an escape. Normal rats will swim around for a substantial period of time looking for an escape route. When they finally figure out that they can't escape, they basically float. What we find in the CMS rats is the CMS rats spend a lot more time immobile. They basically give up much faster, which is an indication of this behavioral despair. What we find in these animals is the opposite of what we saw in schizophrenia patients. A decrease of nearly 50% in the number of neurons firing without changes in firing rate or pattern. Now, what pathway mediates this decrease in dopaminergic neuron firing in depression? Well, studies by Helen Mayberg have identified one area, the subgenual cingulate area 25. This area is hyperactive in sad memory, with tryptophan depletion, which can lead to depression. It shows morphological changes in depression, and any drug that's effective in treating depression brings this hyperactivity back down to normal. SSRIs, normal antidepressant drugs, serotonin norepinephrine reuptake inhibitors, transcranial magnetic stimulation, electroconvulsive therapy, vagal nerve stimulation, deep brain stimulation, even placebos, when they show a behavioral response, restore activity in this area back down to baseline. Now, what happens if we activate the rat equivalent of area 25, the infralimbic prefrontal cortex, in these in normal rats? Well, what we find is just activating the infralimbic prefrontal cortex gives us the same change as the chronic mild stress, a decrease by about half in the number of dopaminergic neurons firing. This decrease in dopaminergic neuron firing produced by activation of the infralimbic prefrontal cortex can be prevented if we inactivate the amygdala. Depression in humans has been associated not only with hyperactivity in the infralimbic prefrontal cortex, but hyperresponsivity of the amygdala. And in fact, if we activate the amygdala directly, we also get a decrease in the number of dopaminergic neurons firing. This suggests that the amygdala is what's actually driving the decrease in dopaminergic neuron activity. With chronic mild stress, what we find, again, is a decrease in the number of dopaminergic neurons firing. This we can also reverse by inactivating the amygdala. Now, how does the amygdala attenuate dopaminergic neuron firing? Well, there are a number of pathways between the amygdala and the dopaminergic system, but one stands out in particular, the ventral pallidum, the area we think is inhibited in schizophrenia. What we find is that if we activate the amygdala to decrease the number of dopaminergic neurons firing, if we block the amygdala glutamatergic input to the pallidum with this glutamatergic blocker kynurenic acid, we prevent the effects of amygdala activation from changing dopaminergic neuron activity. So we believe that when the amygdala is driven, it activates the ventral pallidum, which then decreases dopaminergic neuron firing back down to control, or back below these control levels. Now, is the infralimbic prefrontal cortex involved? Well, again, if we get our chronic mild stress rats, we get a decrease in dopaminergic neuron activity, but if we inactivate the infralimbic prefrontal cortex, we restore it to normal. So basically, we think that hyperactivity in the infralimbic prefrontal cortex is what initiates this cascade of events that leads to a decrease in the number of dopaminergic neurons firing. So in depression, the amygdala through the infralimbic prefrontal cortex is decreasing the number of dopaminergic neurons firing. So stimuli normally associated with reward cause a very strongly attenuated dopamine response, which we think can lead to this anhedonic state. The consequence, again, is this failure to activate the dopaminergic system to rewarding stimuli. So what we have is a dopamine activating circuit that we think plays a role in schizophrenia. But we also have this dopamine inhibitory circuit that we think plays a role in depression. So we have two independent systems that activate or attenuate dopaminergic neuron responsivity. Now, although the amygdala decreases dopamine population activity in depression, we showed earlier on that stress activates the ventral hippocampus, which increases dopamine activity. So we would normally have a balance between the amygdala activity and the subicular activity, even though these areas actually interact with each other. So what changes in depression? Well, to do this, we wanted to use a model that's also an accepted model of depression, but something that we know can work on a much more rapid scale. So what we used is the learned helplessness model of depression, something that was developed by Seligman more than half a century ago. Basically, you place rats in a box that has a barrier, and you expose them to a very mild but inescapable foot shock. And you do this for 1 day. The next day, you give the rats an escape route. And what we find when we use these Wistar Kyoto anxious rats is that half the rats, when they get an escape route, will escape. But half the rats don't escape. They have learned that they are helpless to control their environment in an analogous manner to the way depressed patients would feel helpless to have any control over their environment. So what we find is that half the rats escape, but half the rats are helpless. Now what we did is to try to find a rapid reversal is we used a new novel antidepressant drug, ketamine. Ketamine has been shown in clinical trials to rapidly reverse the symptoms of depression after only a single dose, and this effect can last for weeks. Ketamine turns helpless rats into non-helpless rats. Now, what about the dopaminergic system? Well, interestingly, if we compare our rats, the non-helpless rats show the same number of dopaminergic neurons firing as the control animals. But the helpless rats that got identical treatment show this huge decrease in dopaminergic neuron activity. Giving a dose of ketamine, restores dopaminergic neuron firing in the helpless animals to make them look like the non-helpless animals, just like ketamine reversed the helpless behavior. Now, why are we getting this effect? Well, in the excitatory pathway, this hippocampus to accumbens pathway, normally if we stimulate it at high frequencies, we get a potentiation, specifically LTP, a long-term potentiation in this pathway. However, in the helpless rats, what we find is just the opposite, a depression in this pathway with activation. We see this only in the helpless rats, not in the non-helpless rats. Ketamine will actually take this LTD and restore it to LTP. So whenever you restore behavior, as far as escape behavior, you restore dopaminergic neuron firing. We also restore normal plasticity in this pathway. So what we think is that in the depressed patient, there's an inactivation in the facilitatory half of the pathway, which means the inhibitory path predominates, leading to the depressed state. Now, how would these different systems be related to the unique systems of schizophrenia and depression? Because obviously, schizophrenia isn't the opposite of depression. Well, in order to do this, what we did is to compare the two models, the schizophrenia model and the depressed model. In the schizophrenia model, when we look at which parts of the dopamine system are affected, what we see is that the biggest change tends to be in the lateral aspects of the ventral tegmental area. In contrast, in the depression model, the biggest decreases that we see in the dopaminergic system are in medial and central areas, not in the lateral areas. Now, this is important because these areas have different functions. Studies by Nissa Abderdargham and Mark Laruelle have shown that in the schizophrenia patient, the biggest change in amphetamine-induced dopamine release is in the associative striatum. The associative striatum is the place where the lateral ventral tegmental area dopaminergic neurons project in the rat. So the dopaminergic system that's most hyperactive in the rat is the one that also shows the greatest increase in amphetamine-induced dopamine release in the patient. However, the medial areas, the areas that project to the reward-related ventral medial striatum, are the areas that seem to be decreased in the chronic mild stress model of depression and that are involved in reward-related behavior. So what we have is a differentiation with the schizophrenia dopaminergic system in the associative striatum being hyperactive, but in depression, the reward-related dopaminergic system is hypoactive, which is why these systems can be independent and why we should be able to have comorbid states of schizoaffective disorder or schizophrenia and depression in the same patient. So basically, these studies suggest that in the dopamine system, we have distinct roles in both schizophrenia and depression. In schizophrenia, hyperactivity in the hippocampal excitatory circuit inhibits the pallidum, causing the salience-related dopaminergic neurons to be hyperresponsive to stimuli, leading to misinterpretation of events. And this, we think, is responsible for the hallucinations and delusions in schizophrenia. In contrast, these chronic uncontrollable stressors acting through the amygdala decrease the reward-related dopaminergic activity, leading to these decreased dopamine system functions like decreased motivation, feelings of helplessness, and an inability to derive pleasure from experiences. Therefore, although the dopamine system may form the basis of a number of psychiatric disorders, the pathology is more likely to originate in regions involved in their afferent control. As a consequence, therapy based on restoring normal function in these regions is likely to be more effective than directly manipulating dopamine receptor stimulation. And then I need to thank the people who were involved in doing so much of this work, especially Pauline Belligeon, who was involved in the depression work, as well as Jared Moraines, involved in the depression work, the people involved in the schizophrenia work, Katie Gill, Stan Floresco, Yijuan Du, Dan Lodge, Holly Moore, who developed the MAM model in my lab, and Eric Zimmerman, Ornella Valenti, who is involved in a lot of the stress work, Margarita Behrens, who did the work with the immunocytochemistry, Jim Cook, who worked with us in developing the alpha-5 compound that we found to be so effective. Thank you for your attention, and I'd be happy to answer questions if you have them.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, locus, pardon me if I mispronounce, coeruleus is a main source of norepinephrine in human CNS. It will activate its activity directly or by using selective NE receptors, agonists or antagonists. Will they change DA system improvements?
Coeruleus provides an input into a number of regions, including the ventral hippocampus. In fact, the ventral hippocampus contains some of the strongest norepinephrine inputs into the system. By activating the ventral hippocampus, we think stress will actually lead to hippocampal drive, which is why, if you noticed, in some of these cases, if we actually injected a norepinephrine antagonist, we change the system. So we think stress can activate the ventral hippocampus in order to activate the dopaminergic system, which is seen with acute strong stressors. We also know that stress activates the norepinephrine input into the amygdala, which is responsible for a lot of the anxiety states. So we actually have this interaction of stress that depends on the intensity of the stress, the duration of the stress, and also the timing. Because acute stressors, while activating the dopamine system, withdrawal from very strong or long stressors actually cause this inactivation that we think is responsible for depression.
How could two models of dopaminergic dysfunction that you presented converge and result in anhedonia reward-related dysfunctions in patients with schizophrenia?
Fortunately, I can't read the whole question here. Oh, okay. We haven't actually looked at it. It would be interesting to look at something like the chronic mild stress model in the MAM model and see if we're getting changes. What we think would happen is that we would get a difference between what's happening medially with respect to the reward-related dopamine system and what's happening laterally with respect to the schizophrenia-related dopaminergic system. Because what we think is we're getting a differential control over the medial lateral areas. So we would be able to activate the dopaminergic system in one part, leading to the schizophrenia symptoms, and inactivate it in another part, leading to the depressive symptoms. But doing both in the schizophrenia model would be quite interesting.
We have a few questions regarding your experience presenting virtually for lab roots. I'm sure you've presented at physical conferences many times over the course of your career. How do you compare it to presenting live in a virtual environment?
Well, presenting live in a virtual environment certainly has positive and negative aspects. Unfortunately, I can't look at the audience to see whether I'm losing their interest or not. But on the other hand, it also makes it so that I can reach a number of people that I wouldn't normally be able to reach. People that are in countries where travel is more difficult or which I haven't traveled to, and certainly students who can't afford to attend a lot of the presentations that I would give in other places. So I really enjoy being able to reach a broader audience, especially people who normally wouldn't be able to attend and get this information from such conferences.
Do you think this is the future of education for the community?
Whether something like this can replace standard lectures is a question. Certainly, it's a way of being able to reach a broader audience. But there's something about the one-on-one interactions you can have with a class or with an audience that you can't replicate online. So I think that they both have their own roles and they both have important ways to supplement each other in order to most effectively reach the target audiences without one necessarily being better or less effective than the other, but each playing their own role in order to most effectively educate the public.
Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. Thank you, Dr. Grace, for your participation and for your informative presentation. Today's webcast will be available for on-demand viewing for several months following. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.